All right, so we are going to continue our discussion about infectious immunology of infectious diseases. And what we did in the first part of the class was to sort of try to look at pathogens in four different spaces or subspaces: extracellular and intracellular, and then extracellular and intracellular. They both comprise two subspaces. And then we talked about pathogens and different paths that pathogens can take. And what we want to do now is to look at any of these subspaces and try to understand the details of the immune response there. So what I'm going to do is to start from intracellular space. And for the intracellular space, I'm going to start from the intravesicular part. And I guess in this class, we'll be able to cover this part. We will leave the rest for the next class, I guess. So immune defense against intracellular microbes. It's better to say intracellular pathogens. Starting from the intravesicular part. So intravesicular space is a peculiar space. We have two groups of pathogens in the intracellular space. One group of pathogens are pathogens which normally do not live inside the phagosomes. They are the space that they proliferate and live is extracellular, but out of bad luck, they've been phagocytosed. So it's not part of the life cycle of the pathogen to be inside phagosomes. It's happened to be there, and the pathogen doesn't like it. What happens for these pathogens is that they get phagocytosed. Then we have phagolysosome formation. Phagosomes get merged with lysosomes, and lysosomes have a lot of different ways to kill the pathogen. One of the very basic ways, they have acidic pH. Their pH is quite low, and a lot of microbes are very sensitive to low pH. They die. What else they have? They have something named lysozyme. So lysosome is the organelle. Lysozyme is the set of enzymes which are present inside the organelle. And lysozyme can cut peptidoglycans that are part of the cell of a lot of bacteria. So that's another killing mechanism. They also have proteases. Proteases are enzymes which cleave proteins. And if I have a pathogen, well, I have a lot of proteins. So if you expose me to proteases, it will cleave my proteins and I will basically get damaged. And some more specific mechanisms like some proteins which bind to iron, which is a necessary element for the proliferation of a lot of different pathogens. So lysosomes have many, many different mechanisms, and you can find a lot more molecular detail in the text. So this way of killing is an innate way of killing. It's part of the innate defense. Why? Because it's not specific to the type of the pathogen, and it will not retain a memory of the process. So it's innate mechanism of killing. By the way, this way of killing also has a sort of a different name. It's called oxygen independent killing. Why we have this name in here? Well, it's implied that we have other mechanisms which require oxygen, but these mechanisms are not required exactly. Reactive oxygen species, but the mechanisms that I'm showing you in here, they do not require it. All right. So a lot of microbes are sensitive to lysosome and its contents. Microbes which don't like to be there, they get killed easily. What if I'm dealing with a microbe or a pathogen like a mycobacterium or leishmania, which has developed ways to protect itself against the content of the lysosome? It gets phagocytosed and it likes it to be there. It's part of its life cycle to be inside phagosomes. Lysosomes and phagolysosome formation is not going to kill the pathogen. Let me name these sort of pathogens hard to kill. Okay. By hard to kill, I want to say that it's not impossible to kill it. What's not going to be done by phagolysosome formation? What happens for these pathogens? Well, for these pathogens, although they sort of resist the harsh environment inside the phagolysosome, nonetheless, sometimes some of them might die or some of the antigens that they have might be cleaved by proteases which are present inside lysosomes. So this is a very, very simplified representation, and I'm sure you guys would see this as something very, very simplistic way of looking at it, but it's sufficient for this class. We have another vesicle inside the cells, which has a protein molecule inside it, and that protein molecule is composed of two polypeptide chains. And whenever we have a phagolysosome, that would get merged with the phagolysosome. The antigens which have been cleaved in here will get attached to the surface of that molecule, and that molecule migrates to the cell surface and starts showing the antigen. What is the name of this? Write it down. You must know this. Write it down. Write it down. Don't say it. We have a molecule composed of two polypeptide chains inside a vesicle. That vesicle gets merged, gets connected and merged with the phagolysosome, and then the antigens that we have in here get attached to the surface of that molecule, and that molecule migrates to the cell surface. Migrates. Where is your piece of paper? Migrates to the cell surface. What is that molecule controlling you with? This your name was? Write it down. Write it down. What is the name? Write it down. Your name? Write it down, Sari. Write it down. What kind of news? Good. Okay. Good. That molecule is named MHC. What kind of MHC? This is MHC class 2. Okay? MHC class 2 is the molecule which captures the antigens inside the phagolysosome and brings it to the cell surface. Why does it do that? Because it wants to attract the attention of T cells. Okay? So I have a CD4 T cell in here and a CD8 T cell in here. The CD8 is written below. If I have an antigen at the surface of MHC, and that MHC is MHC class 2, it will tell the CD4 T cells to come. How does it tell them? It's a little different. Chemokines are involved in there. But then the T cell, CD4 T cell starts recognizing it. From now on, this cell is labeled antigen presenting cell. Okay? So the real name could be a macrophage or a dendritic cell. But then the label, I might have a label, okay? The label becomes antigen presenting cell, or the adjective, okay? So this is a process of antigen presentation. What I'm going to show you in here, you guys already know, but I want to make sure that it's all clear. When this CD4 plus T cell sees the antigen at the presence of MHC or at the surface of MHC, it requires a second signal to get fully activated. Write down the name of the signal, this red triangle in here, and then this blue triangle, this blue rectangle in here, which is the receptor for that. Write down the name of these two molecules. Write it down, don't say it. I want to check. This is the cell quiz. Write down the name of the red triangle and the name of the receptor. What is the receptor? No. What is the name of it? Okay, what is the name of the blue triangle, blue rectangle, the receptor for it? Very good. Good. What is the receptor? What is the receptor? You guys should know. That's not good, not good, not good. What is the receptor for it? CD28. This is not good for a master's immunology class. This is not good at all. Okay, see, that is CD28. Okay, B7 is called a co-stimulatory molecule because it helps with the stimulation. The receptor is CD28. This is one of the co-stimulatory molecules. You have many co-stimulatory molecules. This is one of the more famous ones. All right. So if you give the cell these two signals, this is called signal 1, this is called signal 2. Then the T cell wakes up. It's like you guys live in the dormitory. Do you guys have roommates, any of you? So some roommates, when you call their name, they wake up in the morning. Some roommates, when you call their name, it's not sufficient. You need a stronger signal to wake them up. Okay, you need to give them a punch in the flank to wake them up. T cells are like that. You call their names, You give them the antigen for which they have a specific receptors. They say, yeah, okay, yeah, well, but they don't wake up completely. You have to have a co-stimulatory signal. You need to punch something in their flank to make them wake up. When they wake up, they start producing the cytokine, which is interleukin 2. They produce it in an autocrine fashion. Autocrine, paracrine, endocrine. Autocrine, it means that I produce it for myself. When they produce it for themselves, they start proliferating. They enter mitosis, cell proliferation. They make copies of themselves. And these copies of these T cells have exactly the same receptor. It means that it can go around. And by the way, here I have one cell. And these are molecules. Molecules are very small. Cells are very big. And it's not just one MHC with one antigen. It has to be hundreds of MHC molecules with hundreds of antigens. It's not just one B7 with one CD28. It has to be hundreds, all right, for this to get activated. Okay, these T cells, these are CD4 T cells. They have another name. They are called T helper cells because they help. But what kind of help? What kind of help? What is the problem in here? What is the problem in here for which this macrophage needs help? It's hard to kill pathogen. It cannot kill it by itself. So it requires help. Okay? What kind of help this T cell can provide? It can provide different types of help. The help that is required in here, it's a very specific type of help. That specific type of help, in order to be generated, needs these T cells to differentiate to the so-called T helper 1 subtype. Okay, look at it again. This macrophage has eaten something which is hard to kill. It presents its antigens to T cells. The T cells come and proliferate. Then they say, okay, what can we do for you? It sends them a signal. That signal is named interleukin 12. Interleukin 12 tells these T cells you need to differentiate, you need to specialize in producing interferon gamma. T cells which produce interferon gamma are called T helper 1 cells. T helper 1 cells produce interferon gamma. They also do something very interesting. That roommate that you gave her or him a punch, when it wakes up, gives you a punch back. All right? Gives you a punch back. What is this punch back? Write it down. I expect you guys, after several months of being immunology students, to know the names of these molecules. It's very important. If you don't know it, it means that you need to read your textbook again. Okay, what is this molecule which appears at the surface of T cell? And what is the name of the receptor for it? There are many molecules. One of them is more famous. I expect you guys to know. Write it down. If you can't write it down, that's bad news, okay? It's not bad news for me, it's bad news for you. Because that means that you might fail your final exam. What is the name of it? By the way, you can make a sketch on your piece of paper what I'm doing. What is the name of it? Write it down. The question is, when the T cell gets activated, and it differentiates into T helper 1, it does two things. One is to produce interferon gamma. Another thing is to generate a molecule at its surface, this green triangle, that binds to a receptor at the surface of the antigen presenting cell. What is the name of it? This is called CD4 T ligand, and this is called CD4 T. Okay, so let me give you a word of caution. If you don't know these things by now, how many months it has been? It's been several months. I know that there's been war in the middle and you've gone back, but you are supposed to read the text. If you don't know these things, chances are that you will fail the immunology exam. And if you fail this course, you cannot move on to your second semester. What we will do, we will give you another exam. If you fail it again, your program is ended and it's out of our hands. All right? So be careful. You come here, you're doing your work, you spend money for it, you're away from your hometown, all these troubles and stuff. But if you don't, these are the very basics of immunology. The very basics of immunology. All right. If I don't know CD7, CD28, if I don't know CD4 ligand, CD4, it means that I need to go back to the text. You can read it again. All right. All of these things I'm telling you in here, I expect you to know already because you've been supposed to read the textbook. All right. So that's just a word of caution. All right. So what happens now is very interesting. The help is provided in the form of interferon gamma and also this molecule. Now this antigen presenting cell, in which in this case I'm talking about the macrophage because it's a Mycobacterium tuberculosis, feels much stronger. Now it can kill the hard to kill pathogen. How does it happen? The microbicidal activity increases. I need to change these slides a little bit so that you can see it better. Microbicidal. How does it happen? It starts producing something which is extremely toxic. Reactive oxygen species. The most toxic of the ROS is called superoxide. It's an O2 molecule with an extra electron on it. And the rest of the things which are produced by it. Hydrogen peroxide, and what we have down in here, let me make it a bit, bring it down up in here. Sorry, you can see it. Sorry, I should have thought about, I think the recording is kind of, all right. So this is this way. All right. Enhanced microbicidal activity. Why? Because an enzyme gets activated inside, actually at the membrane of the phagolysosome, which produces superoxide. If that enzyme is deficient, the person develops a disease named CGD, chronic granulomatous disease. All right? Superoxide is very toxic. Why? Because it wants to get rid of the electron. Getting rid of the electron, if I give you an electron, it means that I'm an oxidizer. I'm an oxidant. And if I oxidize your proteins, imagine that you're the pathogen, I'm the cell. It will kill the pathogen. What else it produces? It also produces hydrogen peroxide, and this is called hypochlorite. These are together called ROS. Write it down. What does ROS stand for? Write it down. Write it down. Write it down. Write it down. What does ROS stand for? Whenever we say ROS, you need to know this. ROS. Miss Sandrine, what is ROS? Write it down. Good. Write it down. What is ROS? ROS. No, oxygen dependent. No. ROS, there is no D between. What is ROS? Look it up. Look it up. Take your cell phones. Look it up. ROS stands for Miss Sandrine, Sakra, Miss Salma, Miss Rawa. ROS stands for, okay, good. Reactive oxygen species. Reactive oxygen species. We also have RNS, but it's sort of less important. Reactive nitrogen species. All right. ROS is very toxic. That is why this mechanism is called oxygen dependent killing. To differentiate it from oxygen independent killing by lysosomes. All right. Okay. So people who, I'm going to go a bit faster because I have 10 minutes. People who discovered this process. This is an intricate process, by the way. This is called what we call crosstalk. Write it down. Crosstalk. Cellular crosstalk. What is a cellular crosstalk? Two cells are talking to each other. Cellular crosstalk. Cells are talking to each other. How do they talk? by these molecules at their surface and the receptors, as well as the cytokines. All right. This cellular crosstalk, which is very important, was discovered in mice. How was it discovered? We have an infection named cutaneous leishmaniasis. All right, cutaneous leishmaniasis. Exactly. Mosquito bite, leishmania get injected to the subepithelial space, macrophages phagocytose them, and then a lesion appears. What happens for cutaneous leishmaniasis is that there is a lesion, but that lesion is controlled. It doesn't go too big, but the scar remains. Scar remains, all right? So you can have cutaneous leishmaniasis in mice. You inject them, not the letter M in here. You can inject mice with leishmania and then a lesion appears. But that lesion, when you look at it, there are a lot of macrophages and there are a lot of T cells. And those T cells have gone there and helped macrophages kill the leishmania or keep them in check so it doesn't get too big. All right. So people who discovered this process of antigen presenting cells and T cells and interferon gamma and reactive oxygen species, they discovered it in mice. They said that, okay, this is a very important defense process. But then they raised the question. This is the question that you guys as scientists and future immunologists should ask. Imagine that this is an important process. What happens if I deprive the animal from this defense mechanism? Can I take this defense mechanism away from the animal and then check to see what happens after that? Yes. I want to deprive the animal of this process. The way that they did it, they said, okay, what if we remove the CD4 T cells from the body? What if we remove the CD4 T cells from the body? You have different ways to do that. Like I can block the interaction between them, or I can block the production of these cytokines. But the way that they did it was to remove the CD4 T cells from the body of the animal. There is a way to do that. They did that. They generated mice which did not have CD4 T cells. And they injected those mice with leishmania. Their expectation was that, okay, well, these mice don't have CD4 T cells. It means that they would not be able to generate T helper 1 cells. If they cannot generate T helper 1 cells, there would be no interferon gamma. And then these macrophages should have a very hard time to protect against bacteria. The lesion should get much bigger or even extend all over the body and kill the animal. They did that. And what they observed was completely surprising. The lesion got a little bigger, but the mouse was still able to defend itself. They checked the lesion and they saw that, well, there is still quite a bit of ROS there. And there is still quite a bit of interferon gamma there. Even though these animals don't have T helper 1 cells, they discovered something new. They found out that there is another cell which can replicate part of the activity of T helper 1 cells. These were natural killer cells. These cells look like lymphocytes, but they are not lymphocytes. They are part of the innate immune system. They don't have receptors for antigen and MHC, but they circulate inside the body and probably inside tissues. And whenever they see interleukin 12, they start producing interferon gamma. By the way, production of interleukin 12 is not dependent on the presence of T cells. Even though in the sequence of events, I said that, okay, first antigen presentation, then co-stimulation, then interleukin 12. But the production of interleukin 12 is not dependent on the presence of T cells. Whenever a macrophage Phagocytosis is something which is hard to kill, starts producing interferon gamma. So natural killer cells see interleukin 12 and they say, yeah, okay, yeah, you're producing interleukin 12. There is something, you might need help. I don't know what's wrong with you because I don't have the receptor for the antigen at the surface of MHC. But I don't care about that because I'm an innate immune cell. Whoever gives me interleukin 12, I would give him interferon gamma. And this interferon gamma can help this macrophage to some extent to produce reactive oxygen species. It's not as effective. Very good. Very good. In the real set of events, the help from natural killer cells arrives earlier than the help from T helper cells. This is relatively quick. Why is it quick? Yeah, okay, yeah, it's innate, but let's look at it logically. The reason that this is faster is that this cell does not need to proliferate. It does not need to differentiate that much. Proliferation takes time. It needs 2 to 3 days for these T cells to proliferate. This guy doesn't need to proliferate. These guys need to differentiate. This doesn't need much differentiation. So it's quite fast. However, the help that it provides is not as effective as the help that is provided by T helper 1 cells. Why is it not as effective?
Because it doesn't have the specificity of.
Yeah, okay, you're giving me the textbook answer, the logical answer. Why this help arrives fast, but it's less potent? Why? Because these cells proliferate. You have many of them after 2 to 3 days. This doesn't usually proliferate. The second thing, look at this crosstalk, okay? Sometimes you're talking to your friend, she's sitting there, you're talking. That's like cytokine production. Sometimes you're saying something out loud in the ear. That's a very different way. Okay, so these two cells are attached to each other. In addition to cytokines, they have interactions through these molecules. NK cell, the communication is much stronger. For this reason, this help is fast, but it's not as effective. This help comes late, but is much more effective. All right? Summarize 1, 2, 3. Very colorful. This is the first mechanism of defense. Is this innate or adaptive, Miss Zahra? Is this innate or adaptive? This is innate. Then comes this. I have given it a name to make it simpler. I could have called it mechanism 2. I've said NK-mediated macrophage activation. Is this innate or adaptive, Miss Sabrine? Again, this is innate. We don't have lymphocytes. This is, I've called it Th1 mediated macrophage activation. Is this innate or adaptive? It's a combination of innate and adaptive. It's a combination of innate and exactly. All right. Now I have a question for you guys. Think about it and then answer. During this process, I was saying, okay, this is hard to kill. This is hard to kill. This is a mycobacterium. It's hard to kill. Imagine that mechanism 1, I need to change this slide a little bit. Imagine that mechanism 1 was sufficient. All right. Imagine that I'm not dealing with a hard to kill pathogen. It's a regular pathogen which lives outside of the cells. It's phagocytosed and it easily dies after phagolysosome formation. There is no need for ROS or anything else. Here is my question. If it's an easy to kill pathogen and it's killed by mechanism 1, is it still required for its antigens to be presented to T cells? Is it still required for T cells to come there and become, say, T helper 1? Why, yes?
Because we need to create an immunity.
Why do I need an adaptive immunity? It's killed easily by innate cells. Generate memory cells to do what?
Because adapt.
The one that I'm showing you in here is an easy to kill pathogen which is easily killed by phagolysosome formation. Of what use could be the T cell?
The T cells.
Like imagine, okay, like imagine I want to remove this table. It's very hard because it's heavy. I would ask for your help. You come and help me. But I want to lift this cell phone. I can do it easily. Why should I ask for your help?
Because I can do it better than you can do.
You can lift the cell phone better than me.
I have more capability. Yeah.
No, it's not necessary. Not necessary. I'm sorry. I'm not that old. I can move this. Why is the correct answer is that yes, it still needs to present the antigen. T cells are still needed to come here. In a lot of situations, they need to differentiate to T helper 1s. They need to produce interferon gamma. Why? Why do they need to enhance the response next time? Because this is easily. Okay, anything else? The answer is that yes. This process needs to happen regardless of whether it's hard to kill or an easy to kill pathogen. Why? Because this T cell is not going to provide help exclusively for the macrophage. There are a lot of other cells out there which might need the help from T cell. And one of those cells is a B lymphocyte. If it's a pathogen which lives outside of the cells, you have 100,000 things which are outside of the cells. 1,000 of them get phagocytosed and they get killed by the macrophage. How about the rest of them which are still outside there? You need B cells to defend, and B cells would require help from T helper 1 cells. So yes, the T cell still needs to come here. It needs to get differentiated to T helper 1. It needs to start producing interferon gamma. But that interferon gamma is not going to be helpful, for this cell. And it doesn't need to produce reactive oxygen species. How can I say that I don't need you for this interferon gamma to have any effect on the antigen presenting cell? I need to have receptors for interferon gamma. I don't express the receptors for interferon gamma. What would happen if I express the receptors for interferon gamma when I don't need it? Well, reactive oxygen species are very toxic. You shouldn't produce them when you don't need them. You should not produce ROS when you don't need it because ROS is extremely toxic. You should produce it only when you need it. So I presented the antigens to T cells. They're producing interferon gamma, but I don't need to produce ROS. So I would stay away from interferon gamma. I would remove any receptors for interferon gamma that is present at my cell surface. Let the interferon gamma be produced by Th1 cells and go somewhere else where it's required. I don't need it because I don't need to produce ROS. This is an easy to kill pathogen. All right. So this was the process for defense against intracellular pathogens which are inside vesicles. Let me stop the class here because if I want to start intracytosolic, it will take a relatively long time. We need to set another class. Who is the class representative, by the way? You're the class representative. You need to talk to Dr. Mohammadi to give us another 1.5 hour to 2 hour because I need to tell you about the rest of these things. And if I want to do it now, the class would get too long and you're already tired, I guess. So we will talk about it later. Okay. End of class for now.